Hello, I'm Noah Benson, and this talk is on asymmetries around the visual field in human visual cortex by myself, Eline Kupers, Antoine Barbot, Marisa Carrasco, and Jonathan Winawer. The letters shown here resemble an eye chart. Because the visual system magnifies foveal stimuli relative to peripheral stimuli, if you fixate at the central dot of this diagram, the letters near the fixation point will be much easier to see than those in the periphery. We can scale up the size of peripheral letters to match the magnification of the visual system. This phenomenon, in which far eccentricity correlates with low performance, while near eccentricity correlates with high performance, is common across visual tasks. Additionally, visual task performance is not uniform across polar angle. Here, I have scaled the letters to approximately compensate for these polar angle asymmetries. If we zoom in on just the letters U, X, H, P, C circled here, we can see that these differences are substantial, with the H on the horizontal meridian appearing much smaller than the C on the lower vertical meridian, which in turn is smaller than the U on the upper vertical meridian. The behavioral effect of polar angle on visual task performance has been observed for many tasks. Here, we show spatial frequency thresholds for a Gabor orientation discrimination task plotted around the visual field on the left and replotted in terms of distance from the meridians on the right. Note that because we are plotting the spatial frequency thresholds, higher thresholds indicate better performance. Across tasks like this one, performance in the lower visual field is consistently better than performance in the upper visual field. Performance along the horizontal is better than along the vertical, and performance transitions gradually between the vertical and the horizontal meridians. To quantify the asymmetries between the upper and lower visual field, we can compare the differences in performance between these positions with the mean performance. We can define the ratio of the difference to the mean as the vertical meridian asymmetry, or VMA. Additionally, we can combine across the upper and lower visual field and use the same formula in which we compare the performance difference to the mean to calculate the horizontal vertical asymmetry, or HVA. These asymmetries are present in the retina. Shown on the right is a plot of the density of retinal cones in terms of eccentricity. The densities for the upper and lower visual fields are shown in blue and red, whereas the horizontal densities are shown in gray. Clearly, there's a greater density of cones along the horizontal than along the vertical. However, although asymmetries exist in both the cone and retinal ganglion cell counts, an analysis using a computational observer model found that the asymmetries in cone densities are a couple orders of magnitude too small to explain the differences in performance between vertical and horizontal. Retinal ganglion cells are also insufficient. For more details on this modeling work, see our poster number 270 presented by Eline Kupers. A natural next place to look in the visual system is in the cortical magnification and surface areas of early visual cortex V1 and V2. There are several reasons that early visual cortex is a promising next place to look. First of all, V1 comes immediately after the retina and the LGN in the visual hierarchy. Additionally, V1 processes grading and line stimuli, which are generally used to measure these effects. V1's size correlates with the behavioral measures such as acuity and illusion strength in past studies. V1 is also known to have a greater representation of the horizontal than the vertical meridian in terms of surface area. Finally, monocular stimulation does not affect any of these asymmetries we have measured, so cortex is somewhat implicated. To study the allocation of resources in early visual cortex, we used the Human Connectome Project's 7 Tesla Retinotopy dataset, which is a large publicly available MRI dataset of 181 subjects, each of whom participated in approximately 30 minutes of retinotopic mapping experiments. The animation on the right is showing you the polar angle maps from some of the subjects of this dataset. The colors plotted on the cortical surface correspond to the visual field colors in the screen shown in the middle, assuming that the subject fixates at the white dot in the middle of the screen. To explain how we calculated the cortical magnification around the visual field, I will use this single subject as an example. Shown here are the polar angle maps plotted on the subject's mid-gray cortical surface. To make things easier to see, we will start by inflating the brain. All of these analyses were applied to both hemispheres, but to explain them for now, I will focus just on this subject's left hemisphere. With the brain inflated, we can clearly see the V1-V2 boundary outlined here, with the dorsal boundary representing the lower vertical meridian in red, the horizontal meridian represented in yellow-green, and the ventral boundary representing the upper vertical meridian in cyan. Surrounding V1 is V2, whose boundary is shown with the dotted line here. Before we can perform any of the calculations relevant for this project, we need a method of identifying where V1 and V2 are and how they are organized, and this needs to be applicable to all 362 hemispheres. We can begin this process by using the subject's cortical anatomy to make a prediction of the V1-V2 organization. This prediction is shown here. The prediction is plotted by reproducing the grid displayed on the schema of the visual field on the left onto the cortical surface in V1 and V2 in the brain on the right. We can then improve this prediction by warping the model to best fit the measured retinotopic underlay. The process looks like this. Now that we have a good prediction of V1 and V2 organization, we can look at the meridians. To explain this, I'm going to zoom in on just the lower vertical meridian. Here we see the lower vertical meridian boundary of V1 and V2, divided into 5 isoeccentricity bands. To examine cortical magnification around the visual field, we need to divide this region into isopolar angle bands as well, which should be roughly orthogonal to the isoeccentricity bands. Because the method we use to determine the V1-V2 organization and boundary is automated, we wanted to find a method of calculating these isopolar angle bins that would be robust to small errors in the localization of the boundary. Accordingly, we started by calculating the cortical surface distance along the mid-gray surface, away from the lower vertical meridian boundary. These distances are shown here in the dotted lines. We then determined for each side of the boundary independently and for each eccentricity bin independently the distance from the boundary required to reach plus or minus 10 degrees of polar angle. In this way, a small change in the location of the boundary or in the isoeccentricity lines will result in nearly identical ROIs. We can repeat this for a variety of increasing wedge sizes out to plus or minus 50 degrees of polar angle. The ROIs predicted by this method are shown here, with white indicating the plus or minus 10 degree ROIs and red indicating the plus or minus 50 degree ROIs. Note that additionally, we can analyze either the cumulative wedges around the boundary, such as the plus or minus 50 degree cumulative wedge shown here, or we can examine incremental wedges around the boundary, for example, the 20 to 30 degree wedges shown here. We performed these analyses for both the lower vertical meridian shown here and for the upper vertical meridian shown in blue. Additionally, we compared the surface areas around the horizontal meridian of V1 with the V1 portions of the upper and lower vertical meridians as shown here. Here are the results of this analysis. Both plots are showing the surface areas of the cumulative ROIs. The left panel shows horizontal in gray versus vertical in magenta. The surface areas are shown on the y-axis, while the x-axis gives the ROI width around the meridians in terms of polar angle degrees. Clearly, the cumulative horizontal ROIs are larger than the vertical ROIs at all polar angles. In the plot on the right, the upper versus lower ROIs are slightly more nuanced, however. By zooming in on just the plus or minus 10 degree ROI, we can see clearly that the lower vertical meridian ROI in red has a higher surface area than the upper vertical meridian ROI in blue. Note that the colored boxes in these plots are showing the interquartile ranges, and the black lines span 95% of the data. The crosses in the middle of the boxes show the 95% confidence intervals around the mean, so these ROIs are significantly different. However, the asymmetry gradually reduces as we increase the size of the ROI, such that by the time we reach the plus minus 50 degree ROI, the upper and lower visual fields are no longer significantly distinct. We can use these data to plot the incremental asymmetry around the visual field as well. These data are shown in the bottom row with the horizontal vertical asymmetry on the left and the vertical meridian asymmetry on the right. In both cases, the asymmetry lies in the same direction as the behavioral asymmetry and decreases gradually away from the meridians. Here, we have replotted these data on the left while comparing the behavioral measurements shown earlier on the right. Again, the behavioral data shows spatial frequency thresholds, so higher thresholds mean better performance. On the left graph, higher means more surface area. It is clear that the data are qualitatively similar. Both decrease gradually with distance from the horizontal meridian. Finally, shown here is a comparison of asymmetry across the visual hierarchy, starting with the retinal cones, shown as the black line, including the retinal midget ganglion cells, shown as the gray line, and finally the cortex, shown as the dashed line, all across eccentricity values. For both the horizontal vertical and the vertical meridian asymmetry, the asymmetry grows as one goes up the visual hierarchy, from cones to RGCs to cortex. To conclude, asymmetries around the visual field persist throughout the visual hierarchy, and although front-end optics and cones are asymmetric, they explain only a small fraction of behavioral variance. In Cortex, asymmetries in V1 are a good qualitative match to behavioral asymmetries, with both V1 surface area and performance decreasing gradually from the horizontal to the vertical meridians. Additionally, both cortex and behavior have a substantial bias towards the lower versus the upper vertical meridians. Finally, cortical asymmetries enhance retinal asymmetries and are related to behavior. This concludes my talk on asymmetries around the visual field. Thank you for your attention, and please see the following link for further information. Additionally, if you are interested in this topic, posters 116, 270, 873, and 1524 are on similar topics. Thank you.